Hi, everybody. I am Olfat Khannous, a PhD student at the Comparative Genomics Lab at the Barcelona Supercomputing Center, with an affiliation to the Institute for Research in Biomedicine of Barcelona, under the supervision of Professor Toni Cabaldon. And for me, it's a pleasure to introduce here our work on microbiome profiling of fecal immunochemical test samples as a source of biomarkers for colorectal cancer screening. You can find this work recently published in this QR code. So let's start. Colorectal cancer is the third most common cancer and the second leading cause of cancer deaths worldwide, being its incidence a healthcare challenge. It is known that most of the cases of this malignant disease develop from the transformation of normal epithelia to adenomatous polyps that ultimately lead to invasive cancer. And this process is gradual and involves both genetic and environmental factors. Several studies recently suggested a potential role of the microbiome in the origin and development of this disease. Colorectal cancer screening here in Catalonia, as implemented in many regions, is based on the Fecal Immunochemical Test, which is a sample that looks if there is hydrogen blood in the stool. So about this particular threshold, the persons are subjected to colonoscopy. However, as you may know, colonoscopy is invasive, time-consuming, and expensive. And it is known that only around 30% of the cases that are subjected to colonoscopy have a clinically relevant result, so outcome. So there is a real need to reduce this high false positive rate and to the persons that are in the waiting list to subject them before if they have a clinically relevant outcome, as the earlier we detect this cancer, the better prognosis. So the overall objective of this study was to detect before colorectal cancer individuals and individuals of precancerous lesions with clinical relevance, and to reduce the high false positive rate of the current strategies based on fecal immunochemical test by analyzing the microbiome of these samples. So we included around 3,000 individuals that had a FIT positive result and that underwent colonoscopy, and that we got the 16S RNA sequencing. So for those that are not familiar, in the microbiome field, one of the most used strategies to profile the microbiome is the 16S RNA sequencing, which is based on this 16S gene, that it's the small subunit of the ribosome, and that in bacteria and archaea in particular, it's interesting because they have some regions that here are represented in green that are hypervariable, that allow us to distinguish which bacteria we have and in which relative abundances we have them. And also we have these blue regions, so these are conserved regions that allow us to use universal primers to make the amplicons. So in this case, we included both non-clinically relevant individuals and clinically relevant individuals, considering different types of diagnosis across the path from normal epithelia to colorectal cancer. And well, let's talk a bit of the results that we obtained. So we compared our groups of individuals in the different diagnostics groups across this path from normal epithelia to colorectal cancer, detecting differences comparing the different groups. Interestingly, we detected that the most distinct group was colorectal cancer as compared to the other ones. So what we did was to apply our differential analysis, but comparing colorectal cancer versus non-colorectal cancer and clinically relevant versus non-clinically relevant. And we detected some interesting differences. So for instance, in this comparison, we detected Akkermansia muciniphylla with highest effect size. And it is known that in colorectal cancer individuals, there is an overexpression of some mucins. And this is a mucin-degrading bacteria. So maybe we are observing an overrepresentation of this bacteria because of availability of substrate. So taking in consideration these observed differences, we wanted to derive a machine learning classifier that would distinguish which samples would benefit more from a colonoscopy intervention or not. So in this case, and to make the long story short, we end up working with a two-phase machine learning classifier in which our samples are classified before if they are colorectal cancer versus non-colorectal cancer, and the ones classified as non-colorectal cancer are subject to a second classification according if they are clinically relevant versus non-clinically relevant. We are using both the taxa abundances, but also clinical data such as sex, age, and the fit value. So as you may know, in machine learning classification strategies, we normally split our dataset in training and test. And in this case, in order to evaluate the strategy, and in order to avoid lucky splits, what we did was to perform this 100 times, so with 100 random splits. And well, we put our focus on the magnification of the recall, as a false negative is of more medical concern. And you see that we have a good recall, but poor specificity and area under the curve, but still this is significant in this context. And interestingly, we received 100 individuals extra from this screening, that it is a balanced dataset, and we are making some optimization of our classifier. And well, to sum up a bit, the optimal solution that we got so far is based on 4 taxa per phase, plus the clinical variables, and we are detected so the 100% of colorectal cancer individuals, 96% of the clinically relevant individuals, and we are saving 12% of the unnecessary colonoscopies. So all in all, we can sum up this work as that we reconstructed changes in the microbiome profiles of these feed samples associated with different colonoscopy outcomes, and that we developed a machine learning classifier with high sensitivity for colorectal cancer and clinically relevant individuals, while significantly reducing the current false positive rate. Thanks a lot for your attention. I'll be happy to answer any question if you have them.